अस्सलाम वालेकुम फ्रेंड्स, कैसे हैं आप सब लोग। आई होप आप सब लोग बिल्कुल ठीक होंगे। सो वेलकम टू वीएलओजी बाय शोर। अब हम करते हैं अपनी स्टडीज़ को कंटिन्यू। हम लोग पढ़ रहे थे BIF101 इंट्रोडक्शन टू बायोइन्फॉर्मेटिक्स। सो हमारे पास आ गया है यूकैरियोटिक जीनोम। सही है, ये वाला टॉपिक हमने कंप्लीट कर लिया। हम चलते हैं टॉपिक नंबर 22 की तरफ। इसको कर लेते हैं, टेक क्विज करें। तो स्टार्ट करेंगे हम अपना क्विज। इन यूकैरियोटिक एंटिटीज have only operons assisting in gene expression transcription that takes place in the nucleus and translation in the cytoplasm. Transcription occurs in the cytoplasm and translation in the nucleus. In the presence of a CAMP molecule, it carries out protein synthesis. Transcription takes place in the nucleus and translation in the साइटोप्लाज्म। सही है, और ये हो गया करेक्ट। थैंक्स लॉर्ड। अब चलते हैं अपनी वीडियो की तरफ, उसको अच्छे से सुन लेते हैं। आई एम सॉरी, अब मैं वहाँ से ओपन कर देती हूँ।
Now let's see how we can use PCR technology to sequence DNA. I will remind you that genome project, which was basically sequencing human genome, $2.7 billion was spent to sequencing human genome. That's how important it was. It took 13 years to sequence the human genome. Let's see how we can sequence a DNA segment using PCR technology. First of all, let me introduce you to the DNA nucleotides once again. We are familiar with ribonucleosides. We are familiar with the deoxyribonucleosides. Here I am going to introduce you a new type. This is dideoxyribonucleoside. As you know, the carbon atom number 3 has the 3' hydroxyl, which is required for extending the DNA chain. If the 3' hydroxyl is not present, the DNA new nucleotide cannot be added to the growing DNA fragment. So we use the dideoxyribonucleosides in a PCR reaction for sequencing. Let's see how that is done. We make 4 different types of deoxynucleotides, one for each base present in the DNA. And special thing about these dideoxyribonucleosides is that they contain a glowing molecule which is specific to each base. For example, C here is red color, G has blue color, T has green color, A has yellow color. So these dideoxyribonucleosides will always have a molecule attached to it which will emit yellow color. T thymine will have a molecule attached to it which will emit green color, and G blue color, C the red color. So we run our PCR reaction. We have the template DNA. We want to know what is the sequence of this DNA strand. We have a primer. We have little bit information about this DNA fragment. We have to know the little sequence so we can design the primer. The rest of the sequence we need to determine. So we add these components, the normal components for PCR reaction, the normal nucleotides and the enzymes, and additionally we add these special dideoxyribonucleosides attached to their relevant glowing molecules in a very, very small quantity compared to the normal nucleotides. Now when we run the reaction, these bases are incorporated at random. Say for example, we had our primer and it was being extended. Here the primer has annealed. This is the primer sequence. There's the 3' hydroxyl. The next base here, whatever base that is added here would be the base that is complementary to this question mark. So when the base is added, if it is a normal base, it is fine. But if it is one of those special bases that we talked about, dideoxyribonucleotide bases attached to a specific color, the reaction will stop right here. Okay, so we will basically generate a band, DNA band, which is short. Let's just, for the sake of argument, say it was A. So actually, let's just say it was a G. That will make our life a little easier. Let me erase this. So let's just say that it was A. So we had an A that got incorporated, and this A was a special nucleotide. It stopped the DNA chain from the primer from extending any further. And now what happened is that we complete this reaction. This procedure is going on at random. In the next cycle, perhaps this chain will grow all the way up till here, and then the next nucleotide that gets added, say for example G, it will be the special nucleotide. So we will generate PCR products of all the lengths from our starting point of the primer to the last nucleotide. All these bands will be formed with the terminal nucleotide being the special nucleotide. Now when we run the products of this PCR reaction on a gel, the shortest one will come out first. So in this case it was a G that has come out of the gel, this fragment has moved the fastest on the gel, followed by another G, followed by C, T, so on and so forth. So this was the first nucleotide that was added on the primer because it is the shortest one. So as we keep on running our gel, these DNA strands will start moving. This gel is a special type of gel. It is basically a column gel. It is a glass column. At one end of this glass column we have a laser that is going to emit a special light that will make our special deoxyribonucleosides glow, and we have a detector or a camera here that can detect what color was emitted. So the first color that was emitted was blue, and we know where the blue color was attached. Blue color was attached to G. Then again a blue color was detected, that was also G. Then a red, which was C. T was, then the next was green, which was T. So you see, sequentially as we move along, we are determining the sequence of our DNA, which can have very significant consequences. We need to sequence DNA for various purposes. To detect certain type of disorders, to find different type of changes in DNA. So we can sequence DNA using PCR technology. Here I feel pertinent to mention that now things have changed a little bit. As I mentioned at the start of this module, that it took $2.7 billion and 13 years to sequence completely the human genome. Now their technologies have already been discovered, have already been invented, that can accomplish the same feat in about a day or so at about $1,000 per human genome. This is the speed at which technology is moving, and now the day is not very far that we can sequence. When we go to our pathologist for a test, we can also ask them to sequence our genome for us.
So friends, हमारी वीडियो हो गई कंप्लीट. हम चलते हैं अपनी रीडिंग की तरफ. और राइट, आई होप आप लोगों को मेरी आवाज़ सही से आ रही होगी. अगर नहीं सही आ रही होती तो आप लोग मुझे बता दिया करें कि यार मेरी आवाज़ सही नहीं आ रही, क्लियर नहीं आ रही. ठीक है, हम PPTs पर कर लेते हैं डाउनलोड. अब बाकी का काम कर लेते हैं. ठीक है. और ये देखें जनाब, a single stranded DNA fragment is isolated from which for which the base sequence is to be determined, the template. ये जो आपका 4dd NTPs is bound to fluorescent dye, ठीक है. A sample of this unknown DNA is combined with primer DNA polymerase 4D NTPs and the NTPs and the fluorescent fluorescent dideoxy NTPs synthesis begins. ठीक है, सही हो गया. फिर उसके बाद में नंबर 4 पर है, the results are illustrated here by what binds to a T in the unknown strand. If ATP-A is packed up, synthesis stops. A series of fragments of different lengths is made, each ending with a DD-NTP. सही. और उसके बाद है the newly synthesized fragment of various lengths are separated by electrophoresis. सही हो गया. उसके बाद each strand fluoresces a color that identifies the DD-NTP that terminated the strand. The color at the end of each fragment is detected by laser beam. The sequence of newly synthesized strand of DNA can now be decoded. It can now be reduced, ठीक है, and converted to the sequence of template strand. सही हो गया. चलिए अभी ये टॉपिक तो अच्छे से समझ आ गया. अब हम चलते हैं अपने बोनस पॉइंट की तरफ ताकि और भी ज़्यादा बेहतर हो जाए. सी है बोनस पॉइंट में ये सेम चीज़ हो कि तुम इसको नहीं दोहराते थे. DNA सीक्वेंसिंग है, इसमें भी सेम ही है, बिल्कुल सेम है सब कुछ. हमने ये सब पढ़ लिया. ठीक है, कर लेते हैं क्रॉस. और उसके बाद हमारे पास पोस्ट असेसमेंट आती है. पोस्ट असेसमेंट के अंदर हम अपना क्विज ले लेते हैं. डैश इज अंदर पोअरली ट्रांसक्राइब्ड. हेट्रोक्रोमाटिन, क्रोमाटिन, यूक्रोमाटिन, हेट्रोक्रोमाटिन. In eukaryotic entities, transcription occurs in the cytoplasm and translation in the nucleus. Transcription takes place in the nucleus and translation in the cytoplasm. So both are correct. So हम चलते हैं आगे. हम चलते हैं अगले टॉपिक की तरफ. हमारा अगला टॉपिक है, पिछला था DNA सीक्वेंसिंग. और
So far we have discussed that DNA carries the
सो ये वाला टॉपिक का हमारा नंबर है, टॉपिक नंबर है लेसन नंबर 23. तो इसका टॉपिक है जेनेटिक इन्फो कन्वर्जन. ठीक है, तो ये असेसमेंट इसकी नहीं है, कोई बात नहीं. हमारे पास में इसकी है वीडियो. सबसे पहले अपनी वीडियो को अच्छे से सुन लेते हैं, स्टार्ट से लगा लेते हैं. फिर
So far we have discussed that DNA carries the genetic information in the form of nucleotide sequence. How is this information, genetic information, communicated to rest of the cellular machine? How genetic information influences the functioning of a cell? A CD, a compact disc, can contain several songs, but the songs will not play on their own. They require specific machinery to do that. Similarly, DNA also requires certain machinery to be able to communicate its information to the cell and modify or change the cellular function. So happens that people realized that the molecular basis of phenotypes were discovered before it was known that DNA is the genetic material. We will start with the experiments of Archibald Garrett in 1908. He was studying a disease called alkaptonuria or black urine disease. He noticed that there was precedence of children of consanguineous marriage or marriage between two cousins or close relatives. These children had a higher rate of suffering from this particular disorder. So he collected samples, urine samples and blood samples, and discovered that these people were missing a particular enzyme, a protein. The name of that protein is homogenistic acid oxidase. It converts tyrosine into homogenistic acid, and tyrosine in metabolism, tyrosine is converted into homogenistic acid, which is then converted into a harmless product. So this acid basically neutralizes homogenistic acid. Low levels of homogenistic acid can be tolerated, but if this enzyme homogenistic acid oxidase is missing or non-functional, homogenistic acid will accumulate and it will cause damage. So this gentleman was able to conclude, and he was able to link biochemical phenotype of a disease to an abnormal gene and a missing enzyme which was missing in the blood samples. Now let's move on a little bit in time. 1940s, the classic experiment of Beetle and Tatum. These gentlemen were studying a specific type of mold called Neurospora. They basically did experiments. They took this organism, this mold, and they mutated this mold using X-rays. X-rays were used as a mutagen, and that resulted in an altered phenotype. And they were able to associate altered phenotype with an altered enzyme or enzyme activity. So generally Neurospora can grow on minimal media, that is minimal amount of amino acids, carbohydrates, lipids, etc. required for this organism to grow. When they used X-rays to mutate this organism, some of these mutated Neurospora strains, they were unable to grow on minimal media. They had to supplement it. So the term prototrophs, the original eaters, or auxotrophs, the increased eaters, they came up with those terms. So if they realized that if they supplemented the media with certain amino acids or certain chemical compounds, they were able to grow those mutants. Let's look at that. So mutants that were arginine, which required arginine plus the minimal media to grow, they called them arch mutants, and they saw the different versions of arch mutant present in the specimen. So they realized that if arginine is the compound that is missing, let's see if the precursor to arginine can also recover these mutants. So they classified each mutation as affecting one enzyme or another in the metabolic pathway that results in production of arginine in the cell. Wild type, meaning the original strain which did not require any special nutrients to grow, and the mutant cells, they were examined for enzyme and their activities using biochemical assays. The results confirmed that each mutant strain was indeed missing a single active enzyme in the pathway of arginine production. Here is a photograph of their experiment. You can see there are the wild type. It can grow in minimal media in which no supplements, additional supplements are added. So and wild type can grow in even if the additional supplements are added. There's strain number 1 or mutant strain number 1. It could not grow unless and until arginine was provided. Even the products ornithine and citrulline, which are intermediates in arginine biosynthetic pathway, they could not recover these mutants. Other arginine mutants could be recovered not only with arginine but with also citrulline. These mutants could grow if citrulline or arginine, either one of them was provided, but could not grow if they were just given ornithine. They could not grow. The third mutant strain needed ornithine, wasn't provided, they were not able to grow, and citrulline and arginine, they also, if that was these two compounds are also provided, they could grow in these compounds also. So here is the interpretation. If an organism cannot convert a particular compound to another, it presumably lacks an enzyme required for the conversion, and the mutation is the gene that codes for that enzyme. Here you can see there is a precursor molecule which is converted first into ornithine by an enzyme A coded by gene A. Ornithine is then converted into citrulline by enzyme B, and then also citrulline is ultimately converted into arginine by enzyme C. So the interpretation is that if the strain had mutation somewhere here, it could not convert ornithine into citrulline, then we would need either citrulline or arginine to grow these mutant strains. So rest is simple and straightforward. So this experiment was one of the initial experiments that linked change in gene to a function or ability of cells to grow, and connecting the phenotype with the genotype through an enzyme.
So friends, हमारी जो वीडियो है वो हो गई है कंप्लीट. हम चलते हैं अपनी रीडिंग की तरफ. डाउनलोड कर लेते हैं और यहाँ पर जो वो कर लेते हैं. Alkaptonuria black urine. The molecular basis of the phenotype was actually discovered before it was known that DNA was the genetic material. He linked the
let's look at the central dogma of molecular biology. The dogma states basically that DNA contains the information, genetic information, heritable information, which is transmitted from one generation to the next. That information is used to make a molecule of RNA which contains now the same information that DNA contained originally. This RNA can be used to form protein. Once the information has been passed on to proteins, you cannot retrieve that information. And also DNA can replicate itself because we need to have continuity of that information for the next generations. So this dogma raised two serious questions. In eukaryotes, we know the DNA is in the nucleus, whereas proteins are synthesized in the cytoplasm. So how is it that the information, genetic information, is moved from DNA, which is present in the nucleus, to RNA? We know DNA doesn't physically move to the cytoplasm itself. The second question that rose was, what is the relationship between a specific nucleotide sequence of DNA and a specific amino acid sequence in a protein? Basically, how is the language of nucleotides converted or translated into the language of amino acids? Amino acids, as we know, make the proteins which ultimately do the work, or they perform the functions which are coded by DNA to begin with. So Dr. Crick is the same gentleman who, along with Dr. Watson, came up with the structure of DNA. He proposed hypotheses, the messenger hypothesis and transcription. He proposed that RNA molecule forms a complementary copy of one of the DNA strands. This RNA physically then moves to the cytoplasm, where it serves the template. It is still in the form of the nucleotide sequence. It serves as the template which can then be translated into the language of amino acids. Secondly, the second question we had was how the translation takes place. So Dr. Crick proposed an adapter, a molecule that can bind a specific amino acid with one region and can recognize sequence of nucleotides with another region to translate the language of DNA into language of proteins. So adapter would have ability to bind, interact with amino acids, which are the basic building blocks of proteins, and another domain should be able to recognize specific nucleotide sequence on the messenger RNA. So this is the overview. DNA is inside the nucleus. RNA is formed. It moves outside the nucleus. This type of RNA is called the messenger RNA because it is carrying the message of DNA from the nucleus to the cytoplasm. Here RNA encounters ribosomes. We already know that these are the organelles that interact with messenger RNA, and they produce a protein which is coded by messenger RNA, which was originally coded by the DNA. Here I feel pertinent to mention the exceptions to the central dogma. There are viruses that are exception to that rule, DNA containing information. That information getting converted into RNA and ultimately into being translated into protein. For example, influenza and poliovirus, their genetic material is not DNA, it is an RNA molecule. That RNA molecule, when these viruses infect a cell, that RNA molecule makes many copies of itself and also makes RNA that can be translated into viral proteins. Viral proteins are required to contain the nucleic acids which have the genetic information. So viruses, as we know, have proteins and the nucleic acids. The other exception is HIV, human immunodeficiency virus, the virus that causes AIDS, acquired immune deficiency syndrome. This virus, the genetic material of this virus is also an RNA molecule. But this RNA does not get converted into another RNA molecule, or it doesn't get converted. RNA does not copy itself like the influenza virus. This RNA is first converted into DNA. This DNA can integrate into the host genome, and from where, like other genes, are transcribed, converted into RNA and then protein, that can then happen. But first, this RNA virus has to convert its RNA into DNA, and then from that DNA it makes more RNA, and some of that RNA is used to make new proteins. So these are the two exceptions, but ultimately the dogma that DNA contains information, communicates that information to RNA, which gets translated into protein, that pretty much holds.
So हमारी ये वीडियो हो गई है कंप्लीट. हम चलते हैं अपनी रीडिंग की तरफ. फटाफट अपनी रीडिंग कर लेते हैं हम लोग. तो इसको कर लेते हैं डाउनलोड और उसके नीचे आ जाते हैं. हाँ जी जनाब, तो ये है इन्फो फ्लोस फ्रॉम डीएनए टू आरएनए टू प्रोटीन्स. ये ठीक है, इन्फॉर्मेशन होती है डीएनए में, उससे जाती है आरएनए में, उससे जाती है प्रोटीन में. ठीक हो गया, इसका ये मतलब है. इसके बाद हमारे पास में हाउ डज जेनेटिक इन्फॉर्मेशन गेट फ्रॉम द न्यूक्लियस टू द साइटोप्लाज्म. What is the relationship between a specific nucleotide sequence in a DNA and a specific amino acid sequence in a protein? The messenger hypothesis and transcription. RNA molecule forms a complementary copy of one DNA strand moved to cytoplasm serves as a template for protein synthesis. ठीक है. The adapter hypothesis and translation. और adapter molecules binds a specific amino acid with one region and recognizes a specific recognizes a sequence of nucleotides with another region to translate the language of DNA into the language of proteins. सही हो गया. Influenza virus and poliovirus exceptions है. RNA to RNA then to protein. ठीक है. Transcribing from RNA to RNA. ठीक है. और उसके बाद HIV में make a DNA copy of the genome. ठीक होगा. और चल लेते हैं हम आगे. हमारा आ गया है जनाब PPTs हमारे आ गए हैं. ठीक है. द डोगमा. info flows from DNA to RNA to proteins. ठीक है. DNA to RNA to proteins. सही. How does genetic information get from binds a specific amino acid with one region and recognizes the sequence of nucleotide